Oh, okay, I copied the link. Should I distribute them or Miss Hafsa will do it?
Hello, hello, are you there? Am I audible? Hello,
Yes, you are audible.
Afsa, are you there? Miss Afsa, are you there? Miss Notebook, kindly take the description, short description, and move the link as soon as possible. I am just receiving many messages.
Yes, I'm doing it right now.
Hello, hello. Am I audible?
Yes, you were audible.
Miss Hafsa, are you there? Miss Notebook, maybe she is facing some network problems. We have to move forward towards the official orientation program. Thank you so much, everyone, to join us on today's orientation program along with introductory bioinformatics session. So, moving forward to, uh, I have a concern. Uh, Notebook, am I audible?
Yes, you are audible and clear.
Okay. So this is my player uh, to be a main organizing committee of that uh, internship program to 2026. This is Ghulam Murtaza. I am speaking from Pakistan, and by profession, I am a plant biotechnologist and genetic engineer. And currently, I am serving as CEO and president at Vicanova International Alliance for Sciences. We are making collaborations worldwide to building portfolio of students, especially in the domain of life sciences and IT fields, by integration of both fields for research excellence. Uh, yeah, so I really feel proud to lead this phenomenal alliance, empowering individuals to excel in the world of science by supporting progressive research collaborations and specialized scholarship guidance sessions. We inspire to the right spot for those who are seeking guidance on their academic professional journey, especially for those aiming to pursue opportunities, scholarships abroad. This is alliance designed with passion and courage and purpose. As someone who understands the challenges, ambitions of exploring a career in life sciences and IT fields, we founded that alliance to provide personalized guidance resources to empower your dreams, whether you are seeking support in research, to explore collaborations, attending scholarship guidance sessions, and internships, symposiums, seminars, short courses, all things that are related to your professional development. So moving forward to it, I hope I am audible to all the participants. Uh, Here I can just it's just like disappointing. We have the internees around 600, but only 200 join. Like maybe some have some genuine reasons, but mostly that is about the commitment and passion, because we really spend a lot of struggles and efforts to arrange that type of opportunities for the students, especially for young research leaders. So moving forward to it, uh, next is Norfolk. here. So, uh, no workplace give a last reminder in the group of internship. Officially, last reminder. Those will not join. Simple is that we will inspect the students, we will check the attendance, and we will take over the action or not. Those will not attend. Simple is that they will not exist in the group, and they that are not uh, totally against the term and conditions of alliances. So thank you so much everyone for joining us here in our 2 days meeting, in today's class orientation session. We will discuss about your report, like how you will build your report. That will be a showcase of your professional journey within that internship program. Here is a sample of report from our previous internees. And last year, they make their report, and we just share here as a sample. So those students, please check these that report, and moving forward to it, Ms. Notebook will guide you further about the report. And after that, our guest instructor, uh, he will provide a core lecture on introductory bioinformatics. Stay connected till end. Also make your attendance in the Google form. We will share the Google form in the chat box, and you have to make your attendance by Google form, not in the live chat. Okay, just remember that you have to fill the form for your attendance. Thank you so much, everyone, to listen me, and really nice to meet you all. And at the end, we will discuss also your queries, your questions. And it is just my last reminder: don't go to inbox of everyone. That is not totally unethical. And if we are sharing all information into the group, and you are still, you have to at least take patience. Okay, it will be better. So next, don't try to go inbox. We will share all the information into the group, and you have to show the professionalism. Like at the time, 1 or 3 person cannot deal 300 to 400 students. Okay, it's a common sense thing. So you have to at least cooperate with the team, and team will cooperate with you. And yeah, we are sharing the Google form. Now I would like to invite Notebook to please continue.
Thank you for giving me the floor. Kindly ensure if I am audible or not.
Yeah, yeah, you are clearly audible. You can continue.
Thank you for ensuring. So, first of all, Assalamu alaikum and a very good evening to everyone. On behalf of the Vitanova International Alliance for Sciences, I warmly welcome all participants to the Internship Program 2026. We are delighted to see all talented students, researchers, aspiring professionals joining us from different institutions and regions. Your presence today marks the beginning of the valuable learning journey focused on knowledge, skill development, and professional growth and international collaboration as well. First of all, I would like to express my sincere gratitude to our leadership, mentors, trainers, organizing team, and all the participants for making this program possible. Now, I would like to explain the final report requirement. As you can see here, it is a sample report, and if we go there, you are gonna see the name on logo of our organization. There is a picture. You can add it, or you can use another picture which suits your report. And yep, you are gonna give the name, then your serial number, then the program, your university, and the country or the city, so that we know that this report is submitted by you. So this is important information that we need. Then we have the certification. It is a simple description which is designed to show for the certification theme. You can use it or you can modify it as your own. That is also fine. Then we have acknowledgement. You can design your own acknowledgement to sincerely show your gratitude or what you learned and how this internship is helping you throughout this program. Then the table of content. Yes, it is also important. So if you mark these types of stations, that will help you to organize your report. And our mission, our founder's vision, that is all nicely described in our website. So if you go in our website, you will see nicely described our mission and vision. So you can take idea from there. And this is our leadership team. Here we have our respected CEO, then senior vice president, and me, as well as our general secretary. Our ongoing projects are mega ambassadorship projects. You can add it. Then there are different types of programs and different types of initiatives we took, and we successfully completed them. You can add them, and we all these information are all present in our website. So if you visit our website, you will be able to find them all, and you can design them by your own and showing your creativity. Then these are our collaborations. If you add them, it is also a good thing. And this is also our collaboration. And then this is the summary, which is showing the whole internship report. What is this and why you were doing this and what you learned, what is your review. You can add it in this summary section. Then we have this part, which is the session. And here you have to do is prepare a brief report about your experience throughout this one session. So suppose you did first session, you were gonna do the first session today. So you were gonna do what is you will make a report of this session with the small description of our speaker, then date, and uh, the station name. And then it will describe what you learned, then how you approached your work, and simple list of activity that you learned or key takeaways. Then you are gonna post it in the suppose there is a LinkedIn and Facebook profile. So you will post the report that you were doing it in online, such as YouTube. So you will take the screenshot of doing the class, and you will take like 4 to 5 is enough, so you can show your experience on LinkedIn or Facebook. You are gonna post with a description mentioning our organization on Facebook or LinkedIn, and you will share your first session experience. Then you will take the screenshot of that post and add in the last of your session report, like this you can see here. Then you, after attaching them, you will go for a second session. Second session is the same as the first. You will also take the, you can see here there's a screenshot of LinkedIn. You can also use Facebook, and must mention our organization so that our alliance cabinet members, our leadership members, or different types of units that we have, they can support you and they can show you or repost your work so that you can also boost your LinkedIn profile or Facebook profile. That will help you for networking and growing as well. So it will help you both academically as well as for your networking. After publishing the post, it will take the screenshot as I told you and attach it here so that your first station or second station or third station is complete. And after making lots of classes, there will be approximately like 15 classes, and you will take all the classes report and submit it in a combined file in our group after this program is completed. I hope you all understand. And yes, kindly try to make this report original, well organized, so that it reflects your genuine experience. We are interested in your learning journey and expect this program that we are putting you through, right? So the detailed report format submission will be instructed in the group as well so that you can also check or cross check it again. This file will be shared for that purpose. So try to make it before the deadline, and also best wishes for you and all of you. Best wishes for your journey, and I hope that we will be able to see your experience in LinkedIn or Facebook or Instagram. Thank you, everyone.
Thank you so much, Ms. Nupu Akhtar, dedicated uh, leadership community member, uh, head of collaborations from University of Science and Technology, Chittagong, Bangladesh. I really appreciate your efforts to explain the core result about the report, because it is very mandatory. Without that report, we cannot make issuance of your certifications accredited. So, moving forward to it, now I would like to again invite Ms. Nopu Akhtar. Please invite our guest speaker, Mania Ajmal. Mania Ajmal, am I clearly audible to you? Mania
Yes, yes,
Ajmal.
you are audible. Am I audible?
Yeah, you're clearly audible. So, Nupu Akhtar, please introduce herself and also invite her. Thank you.
Thank you so much. Now it is my privilege to introduce our estimated speaker, Ms. Maleeha Ajmal. Ms. Maleeha Ajmal is a researcher at Science NUST and UST School for Interdisciplinary Engineering and Sciences, National University of Science and Technology, and the former Bioinformatics Research Officer at Novatech. Her research expertise lies primarily in next generation sequencing, analysis, and metagenomics, two of the most transformative areas of modern biological research. In addition to her bioinformatics expertise, she has extensive experience in machine learning, deep learning, language model, and computational drug design and vaccine design, reflecting her multidisciplinary approach to solving complex biological problems through computational method. Her remarkable achievements include being the winner of the HEC Innovation Expo and the runner-up of the Health Hackathon 2026 organized by ASAB and UST, highlighting both her research excellence and innovative problem-solving capabilities. Please, uh, a round of applause for her. Welcome to our session, Miss. The floor is yours. Kindly share your presentation.
Okay, thank you so much for introducing, Nurber. Can I share my screen or are you presenting the slides?
Your presentation is already shared, so you can just speak. Our moderator will slide it, like go through it.
Okay, okay, fine, thank you. Good afternoon, everyone, and a very warm welcome to you all. My name is Maleeha Ajmal, as Nurpur has already stated, and I'm a master's student of Bioinformatics at NUST. And it's genuinely an honor to be here with all of you today as a part of Vitanova International Online Internship Program. Looking at this group, researchers, students, professionals from different countries, different backgrounds, all gathered around one shared curiosity that itself what bioinformatics is about. When students first hear the word bioinformatics, many of them immediately think of computers, coding, or complicated softwares. Others think it's simply biology done on a computer. But bioinformatics is much more than that. It actually represents a major shift in the way biological research is conducted nowadays. For centuries, biology was primarily an observational science. Scientists observed organisms, described structures, classified species, and performed experiments. The laboratory was center of biological discovery. Today, laboratories are still very important, but something else has become equally important, that is the data. You can see that a single genome sequencing experiment can produce such enormous amounts of data. Thousands of genomes can be generated, stored, and shared across the world at one time. And researchers can measure the activity of thousands of genes at one time, determine protein structures computationally, analyze entire biological systems using computational methods. So, bioinformatics actually emerged to address the problem of understanding the data. So, if I were to define bioinformatics in one sentence, I would say bioinformatics is the science of extracting biological meaning from biological data using computational approaches. So, in the next few minutes, I want to give you a very real, honest introduction to this field, not the textbook version, but what it actually is, why does it matters, what it can do, and most importantly, how someone like you sitting right here where you are today can become a part of it. So, if I were to say what is bioinformatics, one of the reasons students find bioinformatics interesting it is that it does not belong to a single discipline. It sits at the intersection of several fields. From biology, it borrows the concepts such as genes, proteins, evolution, and cellular processes. From computer science, it borrows algorithms, databases, and programming. From mathematics and statistics, it borrows methods of analyzing patterns and testing hypotheses. So we can say that bioinformatics is an interdisciplinary field and it is both its greatest strength and one of the biggest challenges as well. Bioinformatics education often brings together learners from both biological sciences as well as people from computational backgrounds. Biologists learn to think computationally, whereas computer scientists learn to think biologically. And somewhere in the middle, bioinformatics is born. Now, if we talk about what is biological data, what does that even mean? It actually means the sequences. All have been studying sequences of DNA, RNA, and protein. Bioinformatics help us to analyze these sequences that are the thousands of genes and it measures the expression levels simultaneously. It means the three-dimensional structures of proteins. It means the interactions between thousands of molecules in the cell. It means data from clinical trials, patient records, microbiomes, and evolutionary trees. And if you see the volume of biological data being generated today is very large, it's staggering. Every day, researchers around the world run genomic experiments that produce terabytes of data. And if you can see that no human can read it, no team of human can comprehend that information manually, but a well written algorithm can. This is what bioinformatics is. It actually bridges the gap between raw biological information and meaningful biological knowledge. It actually turns data into discovery. And here's something I love about this field: it is not purely computational and it is not purely biological. You need to understand the biology deeply enough to ask the right questions, and you need to understand the computation well enough to find the right answers. Now let's discuss, why does it even matter? Why do you need to study bioinformatics? So, I want all of you to think of the biggest problems in human health today: cancer, antibiotic resistance, emerging infectious diseases. We all know what a pandemic looks like now. Alzheimer's, Parkinson's, rare genetic disorders, these are the major disorders all the health sciences are working on today. And every single of one of these problems has a biological basis, and every one of them is being tackled right now using bioinformatics. If we talk about cancer, we know that cancer is not one disease, it's hundreds of diseases, each defined by specific mutations in the genome. What does bioinformatics does in cancer? It actually lets us sequence a patient's tumor and compare it to the healthy tissue and identify exactly which genes have gone wrong, which genes have been overexpressed, and which genes have been underexpressed. This is what leads to precision oncology, treating the right patient with the right drug at the right time. If we talk about COVID 19, when SARS-CoV-2 was identified in late 2019, it took only 10 days for Chinese scientists to sequence and publish the full viral genome. And before that, it took a very large time to sequence and analyze the sequence of a single species. But they just sequenced and analyzed it within 10 days, and every vaccine developed after it, the messenger RNA vaccine, the adenoviral vectors, they all started with that sequence. And how did that was analyzed? It was analyzed using bioinformatics. Now, if you talk about drug discovery, it traditionally took 12 to 15 years and over $1 billion for bringing a single drug into market. Bioinformatics, particularly molecular docking and structure based drug design, which is central to this internship as well, is cutting down that dramatically by letting us test thousands of candidate molecules computationally before even stepping into the lab. And if we talk about agriculture and food security, bioinformatics is helping develop disease resistant crops, understand the microbial expression and ecosystems in the soil, track spread of plant pathogen. And in a world where we are facing climate change, that matters enormously. And the more skilled people we have, the better equipped the world is to face these challenges. I think the slides are not visible.
I think there might be a network issue. Kindly give me a moment to
Okay, sure.
share it from my side if it's possible. Thank you.
Hello, am I audible?
Yes, you were audible. The slides need to be shown again.
Yeah, please check. It's working.
Yeah.
Okay, thank you
On which
so much. And
money on which slide you are?
yeah,
Slide
yeah,
number.
you're on the right. I guess the previous slide, the one
Uh,
before this one. Yeah,
NGS.
yeah, yeah.
Okay. Continue,
Thank you
please.
so much. Okay, so now we're going to talk about some of the core subfields and the tools you require for them. And there are some key areas in bioinformatics. You all do not need to be an expert in all of them, but you all need to at least know what different domains in bioinformatics are and what possible opportunities you have in the future. So first, we are going to talk about next generation sequence analysis. So let's start with where most modern bioinformatics begin, the data itself. Next generation sequence, as you all know, it's a technology that changed everything. Before NGS was introduced, it took almost 13 years and $3 billion to sequence a single human genome. And today, it only takes about 1 day and costs under $1,000 to sequence the entire genome. NGS works by breaking DNA into millions of tiny fragments, and then we read them simultaneously using different algorithms and databases, and then we use different computational tools to piece them back together. But here's the thing, the sequencing machine does not give you answers, it gives you raw data, and data consists of billions of short sequence reads in a file. And turning that raw data into biological meaning is entirely a bioinformatics job. If we talk about the NGS pipeline shortly, it starts with quality control. You start with tools like fast FQC to check whether the data is clean enough to start with, and then you filter the data, you remove the cells and the genes that are lowly expressed, then you align your reads to a reference genome using tools like BWA. Then, depending on the biological question, you either do variant calling, that is actually finding where your sample different from the reference by checking the mutations in the sample, or you do expression quantification, that is actually counting how many reads land on each gene, which bridges into transcriptomics later. And finally, you annotate and interpret your results and you see which variants are actually biologically meaningful. Are they in known disease genes? Do they change protein function? So you do perform different metabolic pathways, you perform different KEGG analysis to check if the genes you have identified are involved in the disease or not. So NGS is basically the foundation of genomics, transcriptomics, epigenomics, metagenomics, almost everything in modern biology generates its data through some form of sequencing. If you understand the NGS pipeline, you understand where the data comes from and why the downstream analysis looks the way it does. Now, if we talk about transcriptomics, the genome actually tells you what the gene has, but the transcriptome tells you what the cell is actually doing right now in this moment under specific condition. You know that every cell in your body contains the same genome.
Sorry for interrupting. Is it transcriptomics and RNA sequence slide?
Yeah, yeah, yeah. Okay, thank you.
Yeah.
You know that a neuron and a liver cell have identical DNA, but yet they are completely different, they behave differently, and do completely different jobs. How is this possible? Gene expression. Different cells switch different genes on and off at different times, and measuring that comprehensively across thousands of genes at once is exactly what sequencing does. And if I were to talk about a short overview of how RNA sequence analysis is done, so you take a tumor sample and a healthy sample, you run RNA sequencing on both of the samples, and then the genes that are dramatically more active or the genes that are very less active in the tumor compared to the healthy, those are your candidates. This is actually the workflow of differential gene expression done by DSIC2 or HR, is how researchers identify the drug targets, what should be the drug targets for that disease, understand the disease mechanisms, And then there is a newer, more powerful version that is called single cell RNA-seq. So, how single cell RNA-seq is different from bulk RNA-seq or transcriptomics. So, in bulk RNA-seq, we take the average expressions of genes, but in single RNA-seq, we profile each cell individually. Suddenly, you are not looking at an average, you're looking at a map. You can see that a tumor is not a uniform mass but a collection of different cell populations. Some might be proliferating, some might be invasive, some may be immune cells that are suppressed, some cancer stem cells hiding in the corner. So, this kind of resolution is possible today by single cell RNA-seq. If we move toward machine learning and AI in bioinformatics, that is the third core field, subfield I'm going to discuss today, and that is arguably the most rapidly evolving area in the entire field right now. And I do want to spend a moment here because it is reshaping what is possible in the coming era. Can you please shift the slide? Okay, thank you. So, we know that biology generates enormous, complex, high dimensional data. Thousands of genes, millions of sequence variants, hundreds of thousands of protein structures. And if we talk about traditional statistical methods, they struggle with scale and complexity. But the answer to this is machine learning. In machine learnings, we can handle large amount of data at a very easily and very fastly. And within machine learning, we have two subdomains. We have supervised learning and unsupervised learning. Supervised learning models that consist of random forests, sport vector machines, deep neural networks, they are today being used to classify cancer subtypes from gene expression data, predict which patients will respond to a drug, identify disease causing mutations from whole genome sequences. Supervised learning actually contains data with labels, and we use that data to train our models and to do prediction. While in unsupervised learning, we do not have the labels, we only have the data and we feed the data to the model and it learns the patterns itself. And its examples include different clustering methods like we have dimensional reduction techniques such as UMAP or Disney. These are unsupervised methods. We have graph neural networks for checking the protein interactions, and then we have the large language models. These are all unsupervised learnings. And then there is deep learning. You might all have probably heard of AlphaFold. That is actually the deep minds model that predicted the 3D structure of virtually every protein in the human body that has stumped biology for almost 50 years. That was a deep learning model trained on all the known protein structures, and it is the most dramatic example of AI in biology to date. But it is not the only one. Models are now being used to predict the gene regulation, design new proteins from scratch, and interpret the non coding genome as well. So, if you learn Python, basic Python, and pick up even the basics of skitlearn or PyTorch, you will be able to engage with this area, and the opportunities are enormous. Now, let's move towards structural bioinformatics and drug design. This kind of subfield is basically central to this internship as well, that is structure based drug design. Can we move to the next slide, please? Okay, thank you so much. So, we all know that proteins are not just the sequences of amino acids, they fold into precise three dimensional shape, and that shape is what determines their function. We all know that an enzyme has an active site, a pocket where it binds its substrate and catalyzes a reaction, and a receptor has a binding site where signaling molecules dock. And if you can design a small molecule that can fit into one of those pockets and it has the ability to block that protein, you have a drug. This is like similar to a lock and key model. You can consider the protein as the lock and the drug molecule is the key. Molecular docking is actually the computational simulation of that interaction, and we test whether a candidate molecule fits and how tightly it bounds and what its binding energy looks like. And the workflow of structure based drug design typically goes like this. First of all, you have to identify a target protein that based on the disease you are studying. For example, if you are studying some kind of tumor, you have to know which protein you are going to make the drug against. And then you retrieve its 3D structure from protein data bank, that is the biggest repository of protein structures. And then you prepare the protein computationally. You have to prepare the protein for docking. You define the binding site, you run, then you prepare the ligands. There is a very large repository of ligands and drug molecules online, as well as you can use natural molecules as well. You prepare them, you perform different ADME analysis on them that actually predicts a drug's absorption, distribution, metabolism, excretion, and toxicity computationally before it ever enters a lab. And tools like Swiss ADME, etc., lets you filter the candidates that would fail in clinical trials, and this saves you enormous amounts of money and time. Then you can dock your ligands with your protein using Autodock, Vina, or Pyrex. There are different tools, and then you rank them by predicted binding affinity. And the top candidates then can go towards the wet lab validation. And then you can even perform the molecular dynamics to study the dynamic nature of the protein interacted with that ligand. So what used to take months of manual synthesis and testing can be done in computationally in weeks or days or hours as well. This is the power of this approach, and this is why structure based drug design has become standard practice in pharmaceutical research worldwide today. Can we move on to the next slide? Okay, so lastly, we have multi-omics and system levels integration. So, what actually is multi-omics? So, you all know that no gene, no protein, no molecule works in isolation. This is actually the founding insight of systems biology as well. Modern biology has given us multiple layers of data about the same biological system. The genome tells about you about the genes, the transcriptome tells you about the RNA expression, the proteome tells you about the protein level, the metabolome tells you about the metabolic pathways, the epigenome tells you about how the environment can affect your DNA. Each layer tells you something different. But if we can integrate them and then find how changes at the DNA level can ripple through the protein expression and ultimately to the cellular behavior, this is the challenge of multi-omics analysis. And this is the challenge of multi-omics analysis. And this is very powerful in cancer research as well. If you talk about the Cancer Genome Atlas, for example, TCGA, it contains multi-omics data from thousands of patients across dozens of cancer cell types. We can use it multi-omics data in our research to integrate different types of omics and then generate some results. Researchers use this to find not just what genes are mutated, but how those mutations can change the gene expression as well, how that changes the protein networks, and ultimately they can find what drives the disease so we can have different types of drugs against that disease, or we can formulate some types of treatment. Protein-protein interaction networks you built on using different databases like String, and they can be visualized using tools like Cytoscape. They let you tell how your gene of interest connects to hundreds of different proteins and identify which connections might be targeted. So we can also perform protein interaction networks computationally. We can also perform pathway analysis that constitutes of KGK, EGG pathways. We can perform gene set enrichment analysis. So there is a complete list of different things we can perform on our data. So the system views often reveals things you would never find by looking at one gene at a time. It is a difference between examining a single wire and like how the whole circuit works. So it has wide variety of applications in cancer, in drug therapy, in drug discovery, and also in personalized medicine. And if I just want to tell you about some of the key databases that you need to know before you start bioinformatics. So some of the key databases include NCBI GenDank, that is the world's largest repository of nucleotide sequencing. Then we have UniProt and PDB, both contains comprehensive data of protein sequence and functional information about proteins. Then we have GeoDatabase, that has the gene expression data sets from thousands of studies, and we can use them to do microanalysis, NGS analysis. Then we have TCGA, and it actually constitutes of multi-omics data of the cancer of thousands of patients worldwide. Then we have KEGD database, that actually provides the pathways and genome diseases and drugs. All of these databases are free, public, and they are incredibly rich, and learning to navigate them is one of the first skill you will build in this program. So now let's discuss how to get started. Let me give you a practical roadmap. Okay, how to start. Can we move on the next slide, please? Okay, thank you so much. So you might be wondering that all of this sounds very amazing but also overwhelming. So where do I even begin? So I want to leave you with something very practical. So the step one is to build your foundation. You do not need to be a programmer to start bioinformatics, but you need to be very comfortable with the logic of computation. You need to be comfortable with R and Python. I would suggest you to start with R. It is readable, it is flexible, and it is extraordinary libraries just for biological data. Start there. For statistics, you need to be comfortable with concepts like p-values, multiple testing regression, distribution, regression, which are used constantly. And for this, you have different softwares like SPSS, you have Easy Plus, and different softwares to learn statistical analysis as well. You have Minitab as well. Step two is to learn the databases and the core tools. As a bioinformatician, you must know about the databases or different kind of tools that are required to perform an analysis. So spend time with NCPIs, UniProt, and the PDB. Run your first blast search. Download a protein structure from PDB, open into PyMore. These are free, documentation is excellent, and doing it yourself hands-on will help you learn very fast. And step three is to work on real data. The best way to learn bioinformatics is to pick a biological question and try to answer it with data. Download a dataset from GeoDatabase, try to reproduce the findings of published paper. The process of struggling with real data, dealing with format issues, quality filtering, statistical decisions, they teach you more than any course can actually teach you. And step four is read the literature. Bioinformatics move very fast. Following journals like Nature Methods, Genome Biology, PLOS Computational Biology, and Bioinformatics, they will keep you current and they will keep you updated about what is happening in bioinformatics fields all over the world. And the habit of reading matters more than comprehension at first. This internship is your first step. When you're starting on 13th June, which is tomorrow, you will have access to hands-on bioinformatics and molecular docking training. You will have real tools, real sessions, real learning. You will have access to instructors and structured curriculum. Use it fully, show up, ask questions. The people who get the most out of programs like this are the ones who already know everything, and they are the one who engage most actively. And let me leave you with one thought tonight. We live in the most extraordinary moment in the history of biology. We can read genomes, we can predict protein structures, we can simulate drug binding at the molecular level, we can track viral evolution in real time, we can compare the microbiomes of thousands of peoples to understand what makes us healthy or sick. All of it depends on people who can sit at the intersection of biology and computation and make sense of the data. This is what bioinformaticians do. You are entering this field at the very right time. The tools have never been more powerful, the problems have never been more important, and the bioinformatics community worldwide is one of the most collaborative and open in all of science. Almost every major tool and database is free and it is open source. It reflects the culture that the field actually wants to move fast, and you have room to do a lot of things in this field. And my honest advice to you, do not wait until you are ready. You will never feel ready. Dive into the data, break things, fix things, ask questions that might sound silly. This is how every good bioinformatician I have ever known started. And I am very much looking forward to what all of you are going to discover. And thank you so much for having me today. And I'm happy to take a few questions before we wrap up. And thank you so much.
Uh, Thank you so much, Ms. Maleeha Ajmal, for such an insightful and inspiring session. You just made our participants so much curious to learn about the bioinformatics right now. So we will move on the Q&A session right now. So if you have any questions, you can write in the chat box and I will take them to the miss. The first question we have is, why are hub proteins in protein-protein interaction networks considered attractive drug targets? And what are the risks of targeting them?
Can you repeat the question?
Sure. First was the why are hub proteins in protein-protein interaction networks considered attractive drug targets? And what are the risks of targeting them?
Okay, so hub proteins are actually the connecting proteins, and if you target the hub proteins, there is a very big chance that you are targeting the proteins that are connected to them as well. Because in protein-protein interaction networks, there are tons of proteins with connection that are connected to each other. If you disrupt one protein, the other proteins can be disrupted as well. So if you knock one, you can break different pathways. So if you, for example, a hub protein might interact with 50 to 100 different proteins as well. And if you're blocking or targeting a hub protein, you can disrupt multiple disease pathways at one time. It is very efficient as well. And the biggest risk is that you can target the protein pathway that you do not want to target. So that is one of the biggest challenge in targeting the hub proteins.
Okay, thank you for the great explanation. I hope that our participants got their answer. The another question, which is, why is molecular docking considered a predictive tool rather than a definitive proof of drug efficacy?
Okay, so basically, molecular docking is rigid. In molecular docking, we are considering both the ligand and the protein as rigid. They are static. But our body, that is not static, it is very dynamic. So, when we do molecular docking, we do find the binding affinity between the ligand and the receptor, that is our protein, but we cannot evaluate how that protein is functioning in our body. So that's why we move toward molecular dynamics and we move toward different machine learning algorithms as well. So we can check how that protein and ligand interaction would work in our body as well, because our body is dynamic and it's not static. And molecular docking considers both protein and receptor as static.
Okay, thank you. I hope that the participant got her answer. And we have another question is, can bioinformatics play role in diagnosis of disease?
Yes, of course. Bioinformatics can play a role in diagnosis, prognosis, and treatment of a disease. So, if I can give you an overview of how bioinformatics can help in diagnosis of a disease, for example, if you are tackling a specific type of tumor, say you're working on gastric cancer and you want to make a diagnosis whether a patient has a disease or not, you can take the transcriptomic or genomic data of a cancer patient or a patient with tumor, and you can take a sample of a healthy patient, and then you can run sequencing on both the samples, and then when you do the analysis you do differential gene expression on both the samples, and you can visualize which genes are actually overexpressed or underexpressed in the disease as compared to the samples, and then that's how you can make a diagnosis if certain genes that are reported in literature are involved, are overexpressed or underexpressed, are found in the disease sample. It means that patient has that disease. For example, if we talk about breast cancer, BCR is a very big plays a very big role in the diagnosis of breast cancer. So if you see that PCR is overly expressed, is lowly expressed, you can say that this patient might have breast cancer.
Okay, I hope that she got her answer. And another question we have, which is, do you have any recommended website or newsletter from where we can be updated in bioinformatics regularly?
Okay, yes. So there are different kinds of, research papers. I think research papers are the biggest, thing that you can use to keep updated. There is a lot of biological information that is coming day by day, and I recommend that whichever field you are interested in, you just read as many papers as possible, even if you do not understand them. You just start it and you just start reading and you make sense of that. So there are different research papers that are committed directly to bioinformatics, so I suggest you start with that. And we also have a community. I can share the link of that community. That is a bioinformatics community, and we can ask questions. There is a proper forum where different bioinformatics people ask questions and the related people give answer to that.
Okay, thank you for that. I hope that she or he got the answer. Another question is there. It is saying, what are the biggest challenges currently facing the bioinformatics?
Okay, if I were to talk very honestly, the biggest challenges right now we are facing in bioinformatics is the data. It is not that data is not available, but the data that is freely available, we can use it to learn. But when we are working with machine learning, deep learning, LLMs, we need to have a large amount of data. And that kind of data is not publicly available. People do have it, but it is associated with different companies, different hospitals, and we have to request to access the data. So, in my opinion, right now, the biggest challenge that is bioinformatics is facing today is data. And the second thing is obviously computation as well. Most of the companies and most of the universities do not have enough computation to start different works. For example, if you are dealing with LLMs, you need to have large amount of computation to prepare a chatbot or you perform analysis. You need to have computation. So these are the major drawbacks that we may face when we are studying bioinformatics.
Yep, I also think that as well. So another question we have that how this bioinformatics will be applied in the case of crops.
Okay, so in case of crops, we can use bioinformatics to find different disease resistant fertilizers to develop different, we can study different diseases that are attributed to plants, and we can identify the crops, the genes that are associated with the crops, and then we can use them to make different kinds of drugs against those diseases. For example, we have different crop diseases. In addition, we can incorporate AI and bioinformatics collectively in the field of agriculture to develop different kinds of sensors that actually monitor the pH of the soil, weather conditions, and different types of the conditions that are required for a plant to grow. Okay, can we have the next question?
Sorry for the network disturbance. I hope I'm audible. So kindly ensure if I am audible.
Yes, yes, you are audible.
Okay, thank you. There is another question, which is if we want to study the genetic affinity between two species, those genomes are not available in public database. How can we obtain genomic information using accessible X-ray or DNA mapping material?
Um, that depends on if you can sequence if you have access to that genome of that organism, you can sequence it yourself and then you can analyze it with using various tools and databases. But if most of the genomic sequences are available in NCBI and UniProt, but if the protein structure is not available, you can make it yourself using AlphaFold. And if you do not have the sequence, you can sequence it yourself and you can then use it for different analysis.
Yep. Thank you for answering the question as well. And someone asked that as a final year student of animal science, how does bioinformatics help in animal production?
Bioinformatics can directly help in animal production, but it can help in different kinds of animal sciences. For example, you can use computers and biological data to solve animal problems than we do with old school methods. For example, if we talk about breeding and genetics, uh, we can pick the best animals for breeding, and we can study them using bioinformatics, and we select the genes that are the most prevalent. For example, we can use the software to find the SNPs and markers that are linked to increasing the milk yield or meat quality or egg production, for example. And then we can also use algorithms that can predict which of the animals' kids will be the best even before they are born. So we can also track family trees so that farmers do not accidentally breed different similar organisms. So we can use bioinformatics to study different genomes, and then we can select those markers in the animal breeding, and then we can produce more milk or more meat or
Now I
more
would like to
things
invite
as we can do 50
Marwa.
years ago.
Miss Marwa, please
We can also use
introduce
bioinformatics to
yourself,
find different
society,
diseases in
the
the animals.
different
And for example, if you're talking about bird flu or FMD, we can track
Marwa, please.
different viruses or different diseases, and we can study them, and then we can develop different vaccines against them, just as we do in humans as well. So there are limitless opportunities for all kinds of fields in bioinformatics, and depending upon the field, you can choose what you want to do and how you want to make different amends in the field.
Yep, thank you for your great explanation. Now we will take one last question, which is how can bioinformatics play role in marine environment? Is there not much research on marine ecosystem yet?
Yes, that is right. We do not actually have too much data of the marine animals, but the 95% we know unexplored, and most of the marine life it can't be grown in the lab. So, in the marine field, we can use bioinformatics to uh, discover different hidden marine life metagenomics. For example, you can go towards the ocean and we can collect the metagenomic samples from the ocean, and we can use bioinformatics to study those using tools like blast, and we can do different metagenomic analysis and that, and we can discover different phyla or the species that have not been explored yet by using bioinformatics, by using metagenome analysis. And we can also monitor ocean health, and we can monitor the pollution in the oceans. If we're going toward the environment side in the marine environment, we can see how coral reef or fish stocks can be dying. What How you can use bioinformatics to monitor the health conditions and the pollution as
Yeah,
well.
I was hearing, but right now her voice is not audible.
Sir, her voice is audible.
Sorry,
Can
am
you
I not audible?
can
Yes, you
you
were audible.
Okay.
introduce yourself, dear Muhammad, please?
Oh, sorry, sir is probably having another something. Uh, we can move forward, which is as you described everything so beautifully. I think our participants got all the clear information as well. And your valuable knowledge, practical experience, and guidance have provided excellent foundation of our participants as they begin this internship journey. We sincerely appreciate your time and your effort dedicated to sharing your expertise with us today. Thank you so much, Miss. And
Thanks a lot.
who
I
are
yeah,
questioning
problem.
still, I will
I think
provide you
yeah,
our
I think recently
instructor's
also
email so that you can
recently
ask them on email. And I would request her to provide her LinkedIn account to me so that I can also share it with you guys. So that was it for today. I hope everyone got your first session in a good way and hope for the best. See you in the next class. Till then, stay healthy and stay active. Allah Hafiz.
Thank you so much, and Allah Hafiz.
Allah Hafiz, Miss.
Okay, next Mustafa. Mustafa, please. Hello, hello, hello. Is Nupo here?
Yes, our session is done. I would request you to end this stream.
Okay. Thank you so much, everyone, to listen our first guest speaker, and I hope you learn a lot from this session. And yeah, one last thing, stay connected within the group. Don't connect and don't try to connect on in person because we have a lot of commitments, a lot of work on pending. So we will share all the information in the group. Please cooperate with the team, otherwise we will not as well not cooperate with you. So that is the last message from my side, and wish you good luck for your future journey within that international internship program, bioinformatics and molecular biology. Uh, Ritu, Ritu, can you introduce yourself?